As we jump into the structure of the photosystem, it's important to note that this is totally just a cartoon, only a cartoon. But what it does show is it shows the nature of the light gathering pigments, the way in which they function, and the energetics of the process by which electrons are excited and pass through the components of the ETC in order to enable the production of ATP harnessed from the harnessed light energy. Let's begin with all of the pigments that line the basin, shall we call it, of the photosystem. You can think of the photosystem as being kind of like a sink or a funnel that is literally lined with many, many hundreds of light gathering pigments. These light gathering pigments are of a couple of different types: chlorophyll, or the carotenoids, or the phycobilins, depending upon what kind of organism, oxygenic or anoxygenic, whatever their primary pigment and accessory pigments are. So this is called the antenna complex, this entire basin of pigments that just line that surface. And it's like a solar panel that can really, really expand the network of the ability of the photosystem to absorb light. So we recognize these antenna complexes as gaining and gathering the light energy, and they transfer that light energy from all of these gathering pigments down to the very bottom of the sink, so to speak, the drain. And what happens is these antenna complexes, they actually transfer the energy from throughout via something called excitons, which this might get, I was thinking Matt and Shelly, it might get you guys jazzed. I know you're a little more nerdy when it comes to physics and mathematics, and you might be interested to know that these excitons are basically known as electron hole pairs, and that's the form that the energy takes as it's transferred from all of these light gathering pigments down to the very bottom of the drain area of this basin. And this is called the reaction center chlorophyll. So I just picture light hitting all of these accessory pigments, this like really nature's best solar panel, and just getting transferred like bing, 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 all the way down to the bottom drain of this sink of pigments. And what this does is it excites the reaction center chlorophyll. And, and by that, what I mean is that it makes it have a very negative standard reduction potential, meaning that it really wants to give its electrons to the first electron carrying member of the electron transport chain in this photosystem. So that's what we see is these excited electrons that are getting transferred through the chain. And notice that this picture is just an energetic depiction. As electrons are passed downhill energetically and release that energy, we know that that energy can be used to form a proton motive force. And likewise, we know that that proton motive force can be used to form ATP. This is a great spot to review the different types of phosphorylation. So we've met two of them so far. The method by which ATP gets made when there's a high energy molecule to transfer a phosphoryl group to it, that's substrate level phosphorylation. The method by which ATP gets made when electrons are ripped and stripped off of donuts and passed through an ETC and used to make a PMF and then used to make ATP, remember that's oxphos. And now we meet the very last type of phosphorylation to make ATP. And the difference between it and oxphos is only that the energy comes from light to excite the electrons in the ETC and to make the PMF and to make the ATP. So since light is the source of energy for all of that, you might, and hopefully did, guess that this last type of phosphorylation is photophosphorylation. So we now have it substrate level, number one, oxphos, number two, and photophos, number three, are the types of phosphorylation that can be used to generate ATP. So you might be thinking, well, I wonder where these photosystems reside in a photosynthetic bacterium. If it's a cyanobacterium, an oxygenic phototrope, then it will actually sit in the thylakoids just in the same way as that was seen in the chloroplast. So this is crazy because what it means is that it gives further credence to the endosymbiont theory because yes, in fact, cyanobacteria do contain thylakoids. They don't have chloroplasts, right? They don't have those true membrane-bound organelles, but they do have thylakoids, and they do have the ETCs that span the thylakoid membranes, and that's where they house their antenna complexes and photosynthetic pigments, their photosystems. Now, in green bacteria and purple bacteria, those systems are housed in association with the membrane. So in green bacteria in a place called chlorosomes, but they're highly associated with the membrane. And of course, remember with those, they have slightly different light gathering pigments. I want to take further journey into the photosystems of oxygenic phototrophs. So looking up close and personal with cyanobacteria, those that strip their electrons off of water. So let's look at the two photosystems that exist in oxygenic phototrophs. And the discoverers of these systems were incredibly creative. They called them Photosystem 1 and Photosystem 2. And to boot, they're not even in order. So they're generally shown with the opposite one on the left. So that is Photosystem 2 is on the left versus Photosystem 1 on the right. So not only are they very ineptly named, or at least uncreatively named, but they're also kind of reverse of what we might picture them being. These two photosystems have reaction center chlorophylls that absorb light at two different wavelengths, 680 nanometers versus 700. And so they have slightly different energetics, but that allows them to work really well with one another. And in fact, sometimes both of these photosystems are really, really highly active. At other times, only one of these photosystems is active. And that's actually why the one on the right is called Photosystem 1, is because it's the one that's active all the time, no matter whether the cell needs to synthesize just ATP or whether it needs to synthesize both ATP and NADPH. Photosystem 1 will always be active. So in a cell that only needs to synthesize ATP, then only Photosystem 1 will be called into action. So a cell that's not making any reducing power, it will only use Photosystem 1. And what will happen is that, again, light will hit all of these accessory pigments. It will be effectively transferred down to the reaction center chlorophyll. That will be excited to a high energy state. ETC will run and PMF will form and ATP will be made. But what's cool in this process is that it's cyclic. So this is called cyclic photophosphorylation, because at the end of this whole process, the electron gets passed back to the reaction center chlorophyll, where it gets excited again, where it goes through the ETC, where it makes PMF, where it makes ATP, it gets recycled back again to the reaction center chlorophyll and does the whole thing again. So we recognize this as being a very cyclic process. Let's watch it happen. Here comes the light energy, hits the reaction center chlorophyll, excites that up to high energy, passes it through an ETC, makes a PMF, and that's my cartoon way of showing you that it's making a PMF. It's pumping proton through these members of the ETC and then recycling those back to the reaction center chlorophyll. So a very cyclic process. Now you're probably thinking, I wonder what Photosystem 2 is doing over here. I mean, just hanging out watching Photosystem 1. It's kind of like a city crew. It's like, oh, good job, dude. You're doing great. You're making ATP. But it's not doing a whole lot of anything. So let's see, what are the conditions when Photosystem 2 actually gets galvanized. So what we recognize is that when a cell requires both ATP and reducing power, that is, of course, NADPH, when it needs both of those, then it will run both Photosystem 1 and Photosystem 2. This is a process very fittingly called non-cyclic photophosphorylation. Now let's see why it's non-cyclic, because it must mean that those electrons can't get recycled back to the reaction center chlorophyll of P700. And in fact, that's very true. So let's see what happens to them. Well, light hits this antenna complex, excitons transferred down to the reaction center chlorophyll, it gets excited, negative standard reduction potential, passes through the ETC, and we recognize that NADP plus is the recipient. So, whoa, there's where the electrons went. They got put onto and reduced this cofactor, more specifically this co-substrate, converting it to NADPH. Now, reducing power has effectively been made. So one of the two products that we promised from non-cyclic photophosphorylation has been effectively generated. But check it out. In doing so, those electrons that used to get recycled back to P700, they can't get recycled anymore. They are now part of what is the reducing power that can be used to power those dark reactions. So they can no longer be recycled. So where does P700 go about getting its electrons replenished? Well, that's where Photosystem 2, that was just hanging out, being lazy, comes in. It transfers 2 electrons to that reaction center chlorophyll, and that means that those electrons must have come from somewhere. So let's look at what's going on in Photosystem 2. Light hits, antenna complex, excitons, reaction center chlorophyll, excitement, ETC, and here's where the ATP comes in. The ETC of Photosystem 2 is going to pump protons and to make ATP. So we recognize that it's playing that role in adding that other product. But hang on. What happens when it loses its 2 electrons to Photosystem 1? What does it do now? I mean, it doesn't have any more electrons to excite from the reaction center chlorophyll. So that being the case, it's the P680 has to be able to get electrons from somewhere. And Hannah is probably yelling at the screen by now. She's like, yes, it comes from water. You have to water your plants, right? So we recognize that it strips electrons off of water. That's what makes it an oxygenic phototrope, creating as a byproduct oxygen gas. So there's where those replenishable sources of electrons come, is in stripping and ripping the electrons off of water. So I have a rad model that I just happen to have with me that one of my students made. You're going to love this. It shows both cyclic and non-cyclic photophosphorylation. Okay, this thing is kind of massive, so I'm going to do my best. Okay, on this side, it shows just Photosystem 1 getting into the action. And you can see there in Photosystem 1, you've got the, I love the way that this model was built to show the funnel and the accessory pigments and the reaction center chlorophyll. And then you can see the electrons getting excited through the ETC and getting recycled back in a very cyclic process, so cyclic photophosphorylation. And then on this side, she has non-cyclic. So here on this one, you can see really well how you've got the excitement of electrons, the creation of NADPH with those electrons, the replenishment from Photosystem 2 getting into action finally, and then it producing the PMF and ATP, and then getting its electrons from water over here, so we can actually see that happening and oxygen getting made, oxygenic photosynthesis. So rad model. I'll try to bring this to lab too so that you can check it out in lab. So we're almost done, right? Except that we need to look at the dark reactions of photosynthesis and talk about what's going on there. So in order to set the stage for that, let's go back to our awesome jelly donut burning reaction, combustion, as Cindy would say, that we're going to see if we reverse this reaction, we get oxygenic photosynthesis, the making of sugar, the creation of donuts, the making of glucose. And we notice that if we look at that reaction in reverse, we get a reaction with a delta G that is positive. So in fact, let's write down that delta G, recognizing that for the reverse reaction, it would be positive 686 kilocalorie per mole. Of course, if I can read my chicken scratch there. But we can note that with that positive sign, that this reaction relates to this combustion reaction right here that we see relates to the carbon fixation reaction in the following ways. So the carbon fixation reaction is anabolic rather than being catabolic, right? It's a buildup reaction rather than a breakdown reaction. So anabolic versus catabolic, carbon fixation being anabolic, the combustion of glucose or the breakdown of glucose by chemoheterotrophs being catabolic. We also recognize that this is endergonic versus exergonic. So endergonic, energy consuming, a positive delta G versus exergonic. And we would also recognize that the fixation of carbon is reductive versus oxidative when we talk about the breakdown of glucose. That is, the synthesis of glucose requires reducing power, and we know that that's going to be in the form of NADPH being consumed in those reactions of the synthesis of sugar. So these are the ways in which these compare. So we see in many almost exactly an opposite. We might even also add, in this endergonic versus exergonic, not spontaneous versus spontaneous would be something that we could add to that. So let's leave this behind to look at the dark reactions of photosynthesis, but I want to make sure that everybody understands that these reactions that we're about to show don't only occur in photoautotrophs. Remember that autotroph means self-feeder, and in fact, autotroph quite literally means the ability to convert inorganic CO2 into glucose. So we recognize that not only can photoautotrophs do this, but so too can chemoautotrophs. So our rock eaters can also do the dark reactions of photosynthesis. Now, they get their NADPH and their ATP from something other than a photosystem, right? They get it from the oxidation of these mineral compounds of our rocks or of our inorganics. Okay, let's check out the dark reactions of photosynthesis, aka the Calvin cycle. So the Calvin cycle is an amazingly endergonic pathway. I want to just draw your attention to a couple of things. 18 ATPs put in to this reaction to make, yes, you guessed it, only one sugar. So only one sugar is made from the investment of 18 ATPs. So we recognize that as being a pretty high cost. But then when you realize that there are 12 NADPHs also invested, you start to realize the true cost here. So as we go through a turn of the cycle, in fact, we better go through 6 turns in order to make a 6-carbon sugar, right? So 6 turns in order to actually build that C6 molecule. We're going to talk about 3 phases. The first one, interestingly enough, is just like that in the TCA cycle. It is an incorporation reaction in which a bond is formed between the carbon dioxide and the ribulose 1,5-bisphosphate. This transiently makes a 6-carbon molecule that very quickly and spontaneously breaks down into 2 3-carbon molecules. So we get 12 3-phosphoglycerates. So this first step is called incorporation. So the incorporation of the inorganic CO2. And wait a minute, maybe you're sort of starting to recognize, hey, 3-phosphoglycerate? I think I recognize that. Yeah, that's the product of the 7th step of glycolysis. And that's exactly right. And in fact, what we're going to see now is that the second reaction in the Calvin cycle is identical to the 7th reaction of glycolysis. So we get 3-phosphoglycerate converted to 1,3-bisphosphoglycerate. And hey, remember how high in energy that molecule was? That must mean that there's an investment of ATP to make it. And in fact, right there, 12 ATPs, very endergonic pathway, getting put in to generate, to create, to build 1,3-BPG. Now, guess what? This next step is exactly the opposite of step 6 of glycolysis. So 1,3-BPG uses reducing power to create glyceraldehyde 3-phosphate. So look at the consumption here of 12 NADPHs, a lot of reducing power put in. So note that that's not exactly the opposite of glycolysis, because rather than NADH being used, it's always NADPH, because remember, that is the one that is always consumed in biosynthetic reductive biosyntheses. Now, once we get the formation of 12 molecules of glyceraldehyde 3-phosphate, these can be put together in more steps to create that 1 6-carbon sugar. So glucose, right, can be essentially made from this process. We call these steps reduction and synthesis, very fittingly, synthesis of the sugar finally, and we recognize that the input of reducing power would make us understand why this was also called the reduction step or stage. Turns out that the whole Kelvin cycle is not so simple to track as the TCA cycle was, because note this, it says here many steps. So this is a cop-out in a way. We're just telling you that there's a lot of things that happen before we then get ribulose 5-phosphate, and which in a step that is called regeneration forms the intermediate that reacts again with the CO2. Note that this is also endergonic, another 6 ATP put in this last regeneration step of the Kelvin cycle. Now, what some of you will also note, perhaps John dorks out a little bit with the enzyme that catalyzes this first step of the Kelvin cycle. It's called rubisco for short. And rubisco is the most prevalent enzyme on Earth, and that's a really interesting note about it, but it is partly because it is so inefficient that it is so prevalent. But interestingly, it is the enzyme that does allow for the incorporation of inorganic CO2 into the turns of the Kelvin cycle. We are almost done with our coverage of catabolism and anabolism as they are involved in overall metabolism. But I do want to make a quick note, and this is by far the Cliff's Notes version of anabolism. That is all of these many pathways. And in fact, I think it was maybe Christine and Sam who asked me the other day about the difference between NAD that's pooled in the mitochondrial matrix versus that's in the cytosol. And we were talking about how there are reductive biosynthetic reactions that happen in both compartments in a yeast cell, for example. So let's look at some of the biosynthetic reactions that we can see. We will understand a lot of them. For example, acetyl-CoA can be used to build fatty acid side chains. And eventually, when put together with a glycerol backbone from dihydroxyacetone phosphate or glyceraldehyde 3-phosphate, glycerol backbone, we can make fats. Now there's some nuances to that because of course we're in two different compartments, but eventually those all get to put together to make a fat. We know too that alpha-ketoglutarate can be used to make glutamate. We recognize that pyruvate can get used to make alanine. A lot of intermediates of the PPP can get used to make aromatic amino acids like tyrosine. So a lot of different things that can contribute in to biosynthetic pathways. So I want to take a moment to just summarize those in a very Cliff Note version in your notes. If you want more information, your textbook gives plenty of the plenty when it comes to biosyntheses. So take time if you'd like to get a little bit sexier in your mind, you could go ahead and look at more of the details in there. So we'll just jot down that 2 carbon groups, acetyl groups, are used to make fatty acids, and of course that glycerol backbone comes from the glyceraldehyde 3-phosphate or dihydroxyacetone phosphate, right? Those are in equilibrium with one another, so we can basically name them interchangeably. We recognize too that this is a involved process that requires enzymes that catalyze it. And in fact, there's a wicked cool enzyme that builds fats. It's called acyl carrier protein, and it actually has like an elephant trunk on it where it can take 2 carbon units and reach around this really elongating fatty acid side chain where it can just keep adding, you know, with its kind of elephant arm, just keep adding on and adding on to this elongating molecule. So amino acid synthesis, we recognize that some of those come from glycolysis, some of those intermediates from TCA can be used as precursor metabolites, right, to build and synthesize things like glutamate and aspartate. The pannose phosphate pathway is also giving a lot of precursor metabolites to biosynthesis. We mentioned that aromatic amino acids are made from a lot of the intermediates of pannose phosphate. Synthesis pathways are often regulated, and not surprisingly, by feedback inhibition. So if far too much of the amino acid glutamate has been made, it's likely to feedback inhibit the biosynthetic enzymes early in the pathway. A lot of you did a great job on that question on the exam and understanding the idea of feedback inhibition. Nucleotide synthesis is something that we are not going to cover, but if you take the full year-long biochem, you can look forward to the parts as parts synthesis that goes into the complex construction or anabolism of nucleotides. Pretty crazy involved. That being said, we're going to end for today, and I hope you all have a super, super fantastic Friday. It's Friday, and I hope to see you all again soon on Monday.